for the invitation and the introduction. So today I'm going to share with you an unpublished story on ethnic-related aging rate modification. To this audience, perhaps I don't need to introduce much on aging clocks. Clocks are basically the mathematical models to regress to chronological age or biological age. And then with these clocks, we can calculate a person's aging rate by calculating the difference between predicted age and the actual chronological age. Earlier, we have been using 3D facial images to derive a very accurate clock. So we built clocks using PRSR linear models and convolutional neural networks, the AI models. And also we have trained CNN models based on perceived ages. And using all of this coupled with transcriptome analysis of the same people from the blood, we can then analyze what transcript in the blood can potentially mediate lifestyle's impact on accelerated facial aging or decelerated facial aging. We have also used the 3D facial images to derive a very accurate stroke predictor. And we found that this stroke signal is actually solely coming from the blue channel of the 3D image. However, all of these models are built using Asian populations. So I always get this question, are these clocks or models applicable to other ethnic groups? The answer is, I really don't know. So in 2018, we collected over 1,000 African 3D facial images from Ghana, West Africa, with a collaborator. And here shows how Asian faces and African faces change with age. Essentially, on the z-axis, which is the depth direction, we share almost all the same aging-related signatures. But, and also on the x-axis, they're largely similar as well. But on the y-axis, it's quite different across the two different ethnic groups. Then we combined the 1,000 African images together with 4,500 Asian images to train an ethnic shared aging clock. And in this clock, we can see that, of course, maybe because we have more Asian samples, the prediction of the Asian population has a higher accuracy with an MAD of 3 years, but it's not bad on African either. It's 5 years in terms of mean absolute deviation or error of the model. And in both cases, we can see this model just can, if we look at the embedding of CNN model, the first component and the second component can capture clearly an aging trajectory, indicating the model is indeed learning some features from aging. And another feature we can obviously see from these two trend lines is that compared to Africans, Asians age much slower, much faster on the 3D face contour plot. I should say that in order to remove the interference of the skin color, we did not train the model using any of the color information. So this clock is solely based on the contour, the 3D contour of the image. Next, we asked, what are the physiological parameters associated with accelerated facial aging in both cohorts? We can see in both African and Asian, the most significant feature associated with accelerated aging is blood pressure. And also contributing to the high blood pressure is either visceral fat, measured by some instrument or triglyceride measured by blood test. Unfortunately, we cannot collect the blood samples from African, so we didn't have the blood test data, as well as other blood test, other blood-associated parameters. And on the face, we can see that in both Asian and African, this is, which one is Asian? I think this is Asian, and this left one are Africans. We can see blood pressure have a very similar pattern to the aging pattern. However, these features are just more prominent. Again, on the z-axis, that is the depth, Africans show, both African and Asian show this strong signature of flattened central face compared to the rest of the face when the blood pressure is higher. And also on the x-axis, the features are also very similar, but the features on the y-axis are not so much similar, or even sometimes opposite, especially in the females of Africans. And because we do not have the blood samples from Africans, so we asked, how can we estimate what are the ethnic influence on aging rate using only the Asian population? In order to do that, we derived a measurement called ethnic score, or the similarity of a face to average African face or the average Asian face. So here, basically, we draw a line between the average Asian and average African face. That is a vector. And then we use each face and take the projection of each face 3D image, 3D points to project it onto this line. It's equivalent to a cosine similarity. And using this measurement, we can see that if a face in Asian, it looks more like an African, the aging rate of that person is lower. And in contrast, if an African has a face look more like an Asian, his or her aging rate is higher. So that is just consistent with what we found before. Africans have slower aging rate, right? But this also led us to use only the Asian population to investigate the ethnic score influence. How we do that? Since we have the blood transcriptome from 280 Asian population, Asian individuals, we can then ask what molecules in the Asian blood are mediating the African likeness on the face towards the 3D facial image aging rate. And these mediator genes were derived from a causal inference test, and we call this ethnic mediator genes, or the EMGs. These EMGs, if we look at their functional enrichment, they are enriched for neutrophil degranulation and humoral immune response. Surprisingly, we found that these ECG genes are enriched for ethnic-specific SNPs. The ethnic or divergent SNPs are defined by the FST over 0.1. Usually, FST range from 0 to 1. If it's over 0.1, it implicates there's a significant divergence between the two groups. And we can see also that these SNPs often localize to the enhancer regions. What's surprising is that it's actually very counterintuitive. the more, the stronger the enhancer is, the more SNPs, the more divergent SNP we found on those enhancers. Usually, you would expect the opposite. If this enhancer is highly conserved, it will be less divergent. However, in this case, we found more divergence associated with stronger enhancers. And then we also looked at the transcription factor motif enrichment, and we found that ETS family transcription factor motifs are okay.
Can I just switch to this one quickly?
Hello? It's not working. Hopefully it can last for another 5 minutes. And amazingly, many of the SNPs also localize directly inside the motifs of these transcription factors. Actually, these transcription factors, most of them are regulating the immune response, like FLE, which is a very famous ETS transcription factor. And also, we found P73 motif is enriched. P73 is related to aging and senescence. And many of these SNPs are directly localized in the motifs, indicating that they could potentially modify the motif activity and also the transcript level of the downstream gene. Next, we asked, is this modification on aging rate only limited on the 3D phase, or is it actually can be reflected in the blood as well? So we built another transcriptome clock using this 85 ECG genes. And then we found that the transcriptome clock calculated aging rate, which is called the RNA-seq age diff, is even more significantly negatively correlated with African likeness, indicating that if a person is looking like an African, looks more like an African, then the blood transcriptome will be even younger looking than the 3D phase. Even though our likeness, the African likeness, is solely derived from the 3D phase. So that implicates somehow the selection pressure is not on the face. It's perhaps more directly inside the blood. So what could it be? In order to answer that, we deconvoluted the transcriptome, the bulk RNA-seq transcriptome, to different cell types. And we found that African likeness can actually increase the naive T cell to monocyte ratio. We all know from many, including our own single cell clock analysis, and many other people's work, naive T cell decrease almost linearly with age, and monocyte increase almost linearly with age. These are the most important two immune cell types contributing to the immune cell type clocks, or the immune clocks. It also inhibits the memory B cell to activated dendritic cell ratio. Of course, it also increased the naive T to memory B cell ratio. And thereby, it can delay the 3D facial aging rate. And not surprisingly, the majority of these ECGs can also mediate the changes in these 3 immune cell type ratios. So overall, I'm going to summarize I just told you. We found that African and Asians age similarly on the depths of the 3D phase. Both are strongly influenced by blood pressure, and Africans age slower on the ethnic shared 3D facial aging clock. And African likeness in Asians are correlated with slower aging on the 3D phase, and more so with the blood transcriptome aging. And genes mediating the African likeness impact on aging rates are related to ECM and humoral immunity, and may contribute to differential immune cell type ratio in two ethnic groups. Of course, this we need to validate in the real data from Africa compared to Africans compared to Asian, which implicated from this SNP analysis might be under strong divergent evolutionary selection. With that, I'd like to stop here by thanking the most important person contributing to this work. Is my student Yang Wang, and also this is done in collaboration with Wei Wang's group and his student, Entok. They are from Addis Cowan University in Australia. Thank you.
Thank you, Jackie. We have time for 2-3 questions. So raise your hands. Vadim was the first. Then it's Raghav. Okay, just unsure. And then Vera.
Yeah, thank you. It's very interesting. So 2 quick questions. One, what is the African population? And because it depends on where you study this in the continent. And second is, how does it correspond to the fact that Africans' lifespan is much shorter? So do they age slower but still die?
Right. The African cohort is from Ghana, so it's directly from African. It's not American Africans. The reason is we're a little bit hesitant to use the data from American Africans. That's the only transcriptome data, limited number, though, available. Is that they may have influence from other ethnic groups. But here, this is pretty pure Africans, even though you can also see some divergence in Ghana population as well. And yeah, the lifespan question. I don't know about the lifespan, but I think this aging rate is more related to health span rather than the lifespan, because as you can see, at least from our analysis, is more associated with the immune cell functions and immune cell proportions. So I think this is some selection for the fitness, but not necessarily selection for the lifespan.
Oh. Amazing presentation, by the way. 2 quick questions, just like Vadim. One was, typically within African American populations, we see that there is higher risk of cardiovascular events as well as sort of just generally metabolic syndrome. Does that translate into your Ghana African population as well? And do you see that in the transcriptomic level as compared to the Asians? And what do you think about this just these results translating to epigenetic clocks in general? You talked a lot about transcriptomic clocks. About how does this translate to epigenetic clocks? Any thoughts about that?
Yeah, I'll answer the second question first. Yeah, it would be very interesting to see whether the epigenetics clocks are also modified in these regions. I would see maybe, but currently it's very hard to collect samples directly from Africans. So if you have any connections, I would be happy to collaborate with them. What's the first question?
That within African Americans, we typically see higher rates of
metabolic syndrome. Actually, those are probably due to, I don't know whether this is pure speculation, but what we found was that in Africans, we see a cleaner association of high blood pressure with accelerated aging, indicating the effective size is much higher in African than Asian. That could be genetic, but it could also be because the less intervention in Africans. So Africans, normally they don't know they are hypertensive. None of them are taking hypertension drugs.
Okay, two quick questions. First, I noticed in your initial graph that maybe there was some sort of non-linearity, that there is certain age when really Africans and Asians start to diverge. So is the rate actually maybe slower at some point in Asians and then it picks up? So can you comment on that?
Yeah, in the beginning, there's not much difference. Only it's almost like associated with age. The older people are, the divergence is stronger. Mm-hmm. So maybe it's accumulative.
And then how do you think immune phenotype is connected to the face? Because ECM, I understand, but like how immune system are the same genes affecting?
Yeah. Actually, at this point, I really don't know. For example, when we saw this central ballooning of the face associated with stroke probability, the reviewers kept on asking us, that no matter which journal we went to, they all asked me, what is the medical basis for this? I really don't know, but one day I woke up and said, what the heck? Ask ChatGPT. And then it gives this perfect answer. It says the central ballooning is actually called by a medical term central cyanosis in reference to peripheral cyanosis. It refers to the lack of oxygen in this region, and this region shares the same blood flow to the brain. That's why I said, okay, very smart. So I will probably do the same for this.